Diuretics part 4, potassium sparing diuretics. Another category that is important clinically and academically also, questions are asked in the university exams. So let's understand where this category or these drugs work, act, and how do they produce their effects and what are the toxicities related to them. Before we start the actual discussion, let's recall two important physiological principles that we will be requiring later on. First is the electrochemical gradient. You know, the ions move because of their own concentration gradients from high concentration to low concentration. Ions also move due to the electrochemical gradient. For instance, if there are more number of negative charges lined up on one side compared to the other side, then an anion like chloride will diffuse from high number of negative charges to low number of negative charges, or a cation like potassium can move or will move towards the high number of negative charges. A cation will be, in a sense, attracted towards the high number of negative charges. So this is one principle that should be kept in mind when we discuss this category. And the second small principle is about the sodium potassium pump. Sodium potassium pump binds 3 sodium ions on one side and transports them to the other side, and from the other side it brings only 2 potassium ions to this side, which means whenever this is working, sodium potassium pump, it is an electrogenic pump. That is, it creates negative charges, extra negative charges. I mean, it creates a deficit of positive charges because 3 positive charges are sent to the other side and only 2 are brought to this side. That means deficit of the positive charges, and which means relatively excess negative charges. So sodium potassium pump creates relatively excess negative charges on the side of the membrane, one side of the membrane. So with this in mind, now let us start our discussion. This is potassium sparing diuretics. This category was developed probably after the loop diuretics and thiazide diuretics were already there. So we will be also looking at the fact as to why this category was developed at all. Before that, let's start with the normal physiology. What happens at the collecting duct? Well, collecting duct is the site of action of potassium sparing diuretics, point number 1. So we will be looking at the collecting duct closely. Only 2 to 5% of the sodium chloride is reabsorbed from the collecting duct. Only 2 to 5%. Why do I mention it? Because most of the diuretics, they are exerting their diuretic action by preventing the reabsorption of sodium chloride. So we will be interested to know in knowing which segment is reabsorbing how much sodium chloride. For instance, thick ascending limb of loop of Henle, it reabsorbs 20 to 25% of the filtered sodium chloride. DCT reabsorbs some 5 to 10% of the filtered sodium chloride. And now the collecting duct, only about 2 to 5%, where these potassium sparing diuretics are going to act. So it appears to be relatively weak action, isn't it? I mean, if the loop diuretics are acting here, they have got the potential to block 20 to 25% of the sodium chloride reabsorption. So that much sodium can be lost into the urine, or even thiazide diuretics, 10% sodium chloride reabsorption can be blocked by them. But there is a qualitative difference. This is quantitatively, it is appearing weak, our collecting duct and the action of potassium sparing diuretics, but there is a qualitative difference. Look, these two, the loop diuretics and the thiazide diuretics, they are acting in the earlier parts of the nephron, right? So even if loop diuretic, for instance, blocks this entire sodium chloride reabsorption, 20-25%, still the sodium will be delivered to the later parts of the nephron, from where some of it will be reabsorbed. It can get reabsorbed from the later distal parts and the collecting duct. Same with thiazide diuretics. Sodium is delivered to the next part of the nephron, from where sodium can be reabsorbed. So point is that loop diuretics and thiazide diuretics are not the final deciders, final arbiters as to how much sodium will be finally excreted into the urine, because even though they are blocking the reabsorption in one segment, that sodium can go ahead and can get reabsorbed from the next segment. But the potassium sparing diuretics, they are the final deciders of how much sodium will be finally excreted into the urine. How come? Because they are acting at the collecting duct, which is the final site for sodium chloride reabsorption. So if this potassium sparing diuretic category, these drugs, if they block this entire or whatever amount in the collecting duct, then all of that sodium will be lost into the urine. So they are the final deciders of how much sodium will be actually excreted into the urine, and therefore how much will be the extent of diuresis, because sodium loss is causing water loss, natriuresis, and therefore diuresis. So that's an important qualitative difference between these diuretics and the other categories. They are acting at the final site of sodium chloride reabsorption. Coming to the collecting duct, what all other things that happen here? One is sodium excretion. As I mentioned just now, final sodium excretion into the urine is determined at the collecting duct. Then circulating plasma volume. Yes, they cause loss of water into the urine, and therefore plasma volume is going to decrease. So how much will be the loss of plasma water, etc., is finally decided in the collecting duct. And third, osmolarity. Osmolarity of the urine and thereby the plasma osmolarity is determined at the collecting duct and the movement of ions. Also important is the action of mineralocorticoids. Aldosterone is acting at this site, and it is going to cause sodium reabsorption and potassium secretion, and thereby the loss of potassium into the urine will happen because of the action of aldosterone acting in the collecting duct. So diuretic induced potassium changes and loss of potassium is finally decided in the collecting duct. With this background, now let us understand actual movement of ions and how the things unfold here in the collecting duct. Right. Now, there are two cells in the collecting duct. P cell, that's principal cell, which reabsorbs sodium and secretes potassium. And then the intercalated cell will reabsorb this potassium and secrete the H+. And that H+ can be lost into the urine. Alright. So P cell or principal cell, reabsorption of sodium and secretion of potassium. And then that potassium which is secreted into the lumen can be reabsorbed by the intercalated cell in exchange for the H+, which can be lost into the which will be lost into the urine. This is the basic information about the cells in the collecting duct. Let's go into the details. The principal cell, which causes reabsorption of sodium, secretion of potassium, there are no co-transporters here. We saw in the case of loop diuretics and thiazides, the reabsorption was blocked by blocking the co-transporters. In the case of loop of Henle, there was NKCC. So sodium, potassium, chloride were all transported together in the same direction by the same co-transporter, same carrier protein. In the case of DCT, there was NCC transporter. So there were co-transporters which could be blocked by the loop diuretics or thiazide diuretics. So just bind to one transporter and then that movement of 3 ions or 2 ions can be blocked. That's not the case here. There are no co-transporters here, but there are separate ion channels. There are separate ion channels for sodium reabsorption, then for secretion of potassium. So separate ion channels is one important difference. Second is anions are excluded from the transport. Remember, in the loop diuretics and thiazide diuretics, they were binding to NKCC and NCC. NKCC, there was chloride, anion was there, which was transported along with sodium. NCC, so sodium chloride co-transporter. Chloride was being transported along with sodium with the same transporter. Here, there are separate channels, and there is no channel for chloride as such. There is no chloride transporter present here. So it means chloride is not transported in a transcellular manner. It will be reabsorbed in a paracellular fashion, as we will see later. But anions are excluded from these ion channels. They are for sodium and potassium mainly. Okay. And the third thing that you should know is that sodium reabsorption predominates over the potassium secretion. Let's have a look here. Sodium reabsorption is much more quantitatively as compared to potassium secretion. And what is the reason for that? The reason we discussed at the start of this video, sodium potassium pump. I mean, sodium potassium pump removes 3 sodium ions from inside the cell and sends into the blood, and only 2 potassium ions are brought here, which means by virtue of the activity of this pump, more sodium is reabsorbed as compared to the potassium secretion. Isn't it? I mean, that's the fact we already know. Now, because of this fact that sodium reabsorption is more compared to the potassium secretion, therefore, there is going to be a deficit of positive charges in the lumen. More positive charges removed from the tubular fluid compared to the positive charges that were secreted in the form of potassium. So, deficit of positive charges means relatively excess negative charges. We saw that at the start of the video. Relatively excess negative charges in the lumen, and this is responsible for lumen negative potential. We saw in the case of loop diuretics, there was lumen positive potential. Here, there is lumen negative potential because of this kind of a movement between sodium and potassium. So, a lumen negative potential got created, which is to the tune of 10 to 50 millivolts in minus. Because of this lumen negative potential, as we saw at the start of the video, potassium can be reabsorbed because there are excess negative charges in the lumen. So, chloride being the anion will diffuse out, but in a paracellular pathway. As we saw, there are no channels here and there are no co-transporters. So, potassium, the chloride, I beg your pardon, chloride is reabsorbed in a paracellular pathway, and that too because of the electrochemical gradient. All right. The other thing that happens is, because there are excess negative charges, the anion, I beg your pardon again, potassium, the cation, is more and more pulled towards the negative charges of the lumen. Now, what is the channel for the potassium secretion? Let's add here. For the sodium, the channel is ENaC, epithelial sodium channel. That's for sodium movement through the apical membrane into the cell. And then on the basolateral side, there is sodium potassium pump which will send it out. For the potassium, there is the channel called as ROM-K. ROM-K is renal outer medullary potassium channel. Now, I will make a separate video for this channel alone, this channel was originally described or discovered as the category called as inward rectifying potassium channels. This was the first member of that group. It is an ATP sensitive potassium channel, and via the ROM-K channel potassium is secreted. And why more and more potassium is being secreted? It is because of the fact that there is lumen negative potential, and therefore the cation potassium is driving more and more towards this lumen negative potential. So these are some of the facts that we should note about the collecting duct and its happenings. Now coming to the important aspect, what was the need to develop potassium sparing diuretics? Let's understand that. There were diuretics like loop diuretics, highly efficacious, and thiazide diuretics. But what was happening? What's the common theme? That is, they stopped the reabsorption of sodium chloride, and because of that, more and more sodium is delivered to the distal part of the nephron, the collecting duct. So sodium load for reabsorption is increasing in this part, in the collecting duct, because they blocked the sodium chloride reabsorption upstream somewhere. Upstream means before the collecting duct. That means in the DCT or in the loop of Henle. More and more sodium was delivered to this segment. That means there was increased load for sodium reabsorption, and more the sodium reabsorption load, that means more positive charges driven out, that means more lumen negative potential, and that means more and more secretion of potassium caused by the other diuretics, the loop diuretics, the thiazide diuretics. They were increasing that load of sodium reabsorption, and thereby they were increasing the potassium secretion because of that lumen negative potential getting developed here. So there was a side effect of potassium lost into the urine and resulting in hypokalemia, sometimes dangerous hypokalemias. That was the need that they thought of developing some diuretic which spares the potassium, which does not cause potassium to be lost into the urine, and that's why the potassium sparing diuretics were developed, possibly. Okay, so note a few things. The relationship between sodium delivery into the collecting duct and the resultant secretion of potassium. We saw that just now. If the diuretic is acting upstream, it blocks the sodium reabsorption. More sodium comes here in the collecting duct, so more sodium to be reabsorbed and more potassium secretion happens. Note that relationship. The other point, yes, we saw that also. Diuretics that act upstream of the collecting duct, they increase the sodium delivery here, and therefore they increase the potassium secretion and potassium loss. Also note, now this is going to be little more important and complicated. If sodium delivery is associated with an anion, obviously it will be the case. But if it is the anion other than chloride, like say bicarbonate for instance, then it will further increase the lumen negative potential. Yes, note this point here, that sodium reabsorption predominating over the potassium secretion, therefore excess negative charges created in the lumen, and we call it lumen negative potential. But since chloride was getting reabsorbed and removed from the lumen, this lumen negative potential will diminish somewhat, isn't it? Chloride is a negatively charged ion and it is moving out. In a paracellular way, it is getting reabsorbed. So some negative charges are going out. Very good, fine. But then what happens in the case of other diuretics? That they deliver sodium and they deliver some other anion associated with sodium. So that some other anion is not getting readily reabsorbed like chloride. Imagine, chloride is getting removed from the lumen. But if some other anion like bicarbonate, then it does not get reabsorbed, which means what? it will further line up in the lumen, and it being anion, it will further exaggerate that lumen negative potential. Yes, that is what also was happening with the other categories of diuretics. That other anion, instead of getting reabsorbed, it just simply lines up and it adds up to that lumen negative potential. And which means what? Which means further potassium dragged into the lumen by that electrochemical gradient and further loss of potassium into the urine. So this point again important and to be noted, that thereby potassium secretion and excretion will further increase. And also note, yes, with the other diuretics, what was happening is that they cause diuresis and therefore loss of plasma water into the urine. That means decreased circulating plasma volume. And if there is a volume depletion, then there is a role for aldosterone. You already know that renin-angiotensin-aldosterone system, RAAS. So diuretic causing loss of water, causing volume depletion of the plasma will cause aldosterone to come into action. If aldosterone comes into action, what is the job of aldosterone? It further increases the sodium reabsorption from the collecting duct. So diuretics, other diuretics, why they were causing so much potassium depletion? These are the reasons. This is the final one, that the diuretic causes volume depletion. So aldosterone comes into action. It increases the sodium reabsorption, and that means further lumen negative potential, as we saw, and that means further potassium dragged towards it into the lumen and further potassium loss into the urine by the action of aldosterone. So this is the basis for diuretic induced potassium depletion, and that probably was the need that they had to bring in another category, invent another category, the potassium sparing diuretics, that they would not cause potassium depletion, because you know hypokalemia, if serious, it can have serious consequences. So this is the background as to why this category was developed and what was the need for it. With that in mind, before we talk about the spironolactone and other potassium sparing diuretics, let's just quickly understand the collecting duct and the aldosterone action, because the aldosterone action is anyways going to happen and it is going to exaggerate that lumen negative potential and loss of potassium. So we must understand it here. Okay, sodium reabsorption, that's the primary action of aldosterone. And sodium reabsorption occurs in a two-step process. So in the apical membrane, there is this channel called as ENaC, epithelial sodium channel. Via this channel, the sodium enters the cell from the lumen, from the tubular fluids. Sodium enters the lining cell. And on the other side, of course, there is sodium potassium pump, which is the common feature everywhere in the nephron. So sodium will be removed from inside the cell and will be sent out in the interstitium. Finally, it will enter the blood. This is how sodium is reabsorbed. We all know that. What is the action of aldosterone? Aldosterone increases this sodium reabsorption. How? Interesting to see that the aldosterone is acting on both these transporters. Obviously, only then the sodium reabsorption can increase. So what aldosterone does is that on the basolateral membrane there is sodium potassium pump. Aldosterone simply increases the number of sodium potassium pumps on the basolateral membrane. You know, aldosterone has nuclear receptor, so it can cause protein synthesis, and therefore it can synthesize new sodium potassium pumps. Number increases here. On the apical membrane there is ENaC. What you need to know about the membrane proteins is that when their lifetime is done, means their certain lifespan is ending, then they are removed from the membrane and degraded inside the cell. For the removal of these membrane proteins, any membrane proteins, for the removal, first they have to be tagged. and then they are removed and then they are destroyed. Tagging occurs by a system of enzymes called as ubiquitin. Ubiquitin system tags the membrane proteins, any membrane proteins, and then the membrane protein is removed and its lifespan is over. Aldosterone deubiquinates these ENaCs. Aldosterone does not allow ubiquitin tagging of these transporters, these ENaC. It means what? It means ENaC will not be removed that fast, that early from the membrane. ENaCs will stay in the apical membrane for longer durations. So more ENaCs staying for longer durations and more sodium potassium pump. That is how aldosterone achieves the increased sodium reabsorption. Why we need to understand this is because of our category, potassium sparing diuretics. They are going to act against the aldosterone. Let's understand this part now. Potassium sparing diuretics, they are of two types. Well, final bottom line is the same. Decrease the sodium reabsorption. Yes, obviously, I mean, all of the diuretics, they are reducing the sodium reabsorption and thereby sodium loss and water loss. That's common everywhere. How do these act? Well, these act in two ways. One category is aldosterone antagonists. So simply, they will block the action of aldosterone in the collecting duct, and therefore the sodium reabsorption will stop. Sodium reabsorption which was happening under the influence of aldosterone, that will be stopped or blocked, inhibited. So the drug is spironolactone, or there are specific ENaC blockers. Instead of being aldosterone antagonist, which will stop entire aldosterone action, only blocking of ENaC, epithelial sodium channel. So amiloride, triamterene, these are ENaC blockers. And therefore ENaC, epithelial sodium channel, is also called as amiloride sensitive channel. It's going to block that channel. So that is how they stop the reabsorption of sodium, and thereby they also decrease the secretion of potassium. We have seen so far that sodium reabsorption is at the core of the entire thing. If the sodium is getting reabsorbed, it is creating that lumen negative potential. That potential is pulling the potassium out via ROM-K channels, and that potassium is getting lost into the urine. That was the basis for hypokalemia. Now that is not happening. That means potassium loss is not going to happen. That means potassium is spared from being lost. It's not lost. And that's the basis why we call it potassium sparing diuretics. So this is in the principal cell, and what happens in the intercalated cell, which is just next. If there is less of potassium secretion by the principal cell, then that means there is less potassium load for reabsorption. And that means, I mean, this intercalated cell will have less load for potassium reabsorption. And that means less secretion of H+, which is happening in exchange. So H+ secretion also is less by just blocking the ENaC or by just blocking the sodium reabsorption. H+ secretion is less. That means H+ is retained in the plasma, in the body. H+ retained more and more in the plasma, in the body, results in metabolic acidosis. So one important side effect of these diuretics is metabolic acidosis, H+ retention. All right. Finally, let's see the indications. Of course, the primary indications are wherever there is increased aldosterone effect. So primary aldosteronism, primary hyperaldosteronism, it occurs in the Con's syndrome or the conditions like ectopic ACTH secretion. You know, there are tumors which secrete ACTH, and it's the ectopic ACTH secretion, and this excess ACTH will cause excess secretion of aldosterone. So the conditions of aldosterone excess, that is where you give potassium sparing diuretics. They will block the aldosterone action. Simple. Or secondary aldosteronism, like it is associated with the, let's say, heart failure or hepatic cirrhosis or nephrotic syndrome. There is secondary hyperaldosteronism in these conditions. That is where also we can use potassium sparing diuretics, because they will act as aldosterone antagonists and will reduce the symptoms of hyperaldosteronism. Toxicity, this is simple now. Hyperkalemia, obviously, they stop the potassium secretion. So potassium is retained in the body, and potassium retained in the plasma means hyperkalemia will be the result. Another one, hyperchloremic metabolic acidosis. We saw that potassium reabsorption load is less, so H plus secretion load is less. So H plus is retained in the body just like potassium, and therefore there is metabolic acidosis. And another side effects like renal stones and gynecomastia. Spironolactone has been found to produce this peculiar side effect of gynecomastia. Other such drugs like cimetidine, H2 blockers have been shown to have this side effect. So this, in a nutshell, in summary, the potassium sparing diuretics, their actions and physiologic basis of their actions.